how does your body make plasmalogens? And if the body's not making enough of them, how can plasmalogens be restored? So the story about making plasmalogens is actually a really interesting story because it's a very strange thing your body does to make these plasmalogens. So plasmalogens are made in a specific organelle of your cells, of your liver, and in your muscles called peroxisomes. And this is what your body has to do to make a plasmalogen. So first, you eat food, lipids, fat, salad oils, and your body has what's called fatty acid elongation and desaturation enzymes in the cell. And so these C18 fatty acids that we get primarily in our diet, they'll get extended in the body. It'll go from C18 to C20 to C22 to C24, long-chain fatty acids. And once you reach a long-chain fatty acid state, those long-chain fatty acids get transported into the peroxisome, which is a special organelle, and it's called peroxisomes because it makes peroxide. It's a highly oxidative organelle. And then the peroxisome does something called beta oxidation. It takes this long-chain fatty acid and it starts chopping it down. So it chops C24 or C26 fatty acid, goes chop, 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 all the way down to a C8 fatty acid, and then it converts that C8 fatty acid, and no other phospholipid does this, by the way. It takes the C8 fatty acid, converts it to a fatty alcohol, and then it rebuilds it back up again to a 16 or an 18 carbon unit. And then it takes that alcohol and it attaches it to the SN1 position of a glycerol backbone. This is all done in the peroxisome. And each of these beta oxidation steps creates peroxide, which is called peroxisome. And so it's very oxidatively demanding. And this is what your body does to make one single plasmalogen. Then it pumps it out of the peroxisome, and then the endoplasmic reticulum goes to regular phospholipid metabolism. And so the incredible amount of work is involved in making it. The other cool thing is that when the peroxisome chops down the long-chain fatty acids, the acetyl-CoA that's created is what's used to create cholesterol in your body. So your peroxisome makes most of your cholesterol as well. So anyways, so all this work goes on to make these plasmalogens. And they're only in the peroxisome. The peroxisome is an inherently weak organelle. There are strange, there are genetic diseases in children that have peroxisomal biosynthesis disorder. These are highly high mortality diseases. The most pure plasmalogen deficiency disease is called rhizomelic chondrodysplasia punctata, RCDP. These are children that are born with the inability to make plasmalogens. They have very short limbs, very short life expectancies. Most of them don't live to age 9 or 10. And so plasmalogen deficiency is very, very dangerous. And other things can damage your peroxisome as you get older, and we believe that fundamental peroxisomal deficiency. So the strange thing about it is it's almost like a planned obsolescence of the human body. Something so critical that makes up 20% of your neuronal brain density, it's required for the sinus rhythms of your heart, it requires this very sophisticated, unique biochemical system in your body. So now the good news is that it's actually relatively easy to manufacture this ether lipid backbone. And so we've done that, and there's different types of precursors. So we manufacture what's called bioprecursors. So that's how your body makes it. So you can, you can stimulate plasmalogen biosynthesis through exercise, specifically resistance training, muscular training will help your body make plasmalogens. But that's typically not sufficient to reach the highest levels that you need. So the ability to, how can we restore plasmalogens? We can restore plasmalogens very easily by delivering to the body the whole plasmalogen molecule that the peroxisome makes just before it goes in the endoplasmic reticulum. So we can provide the ether lipid backbone, and what that does is there's two things. One, it prevents the ability to, you don't have to manufacture as much, but then the body can take that bioprecursor and use it to make all the other plasmalogens of the body. Because once you get that ether bond in place, the natural biochemistry of the body can take it from there and get elevated levels. And what we found is that we can specifically target types of plasmalogens. Like we can put a DHA plasmalogen, which is specifically important for neuronal health, versus a oleic acid plasmalogen, which is involved primarily in white matter myelin sheaths. So we can target different plasmalogens, even though they generally will redistribute. You can kind of somewhat target the different types of plasmalogens. So restoring plasmalogens is actually fairly straightforward, and that's why the clinical trial is pretty exciting, because we're dealing with a very natural, safe way of restoring and augmenting plasmalogen levels, and we're using the natural human biochemistry of the body to do it.